mitochondria. So, mitochondria are one of the very important cell organelles of an eukaryotic organism. So, these are present only in the eukaryotic cells and absent in prokaryotes like in PPLO, that is pleuropneumonia like organisms, and bacteria like organisms. So, these mitochondria are mainly concerned with the Krebs cycle and oxidative phosphorylation. The name of this mitochondria was derived from Greek language, in which mitos means thread and chondrion means grain. So, which is showing nearly a thread like structure, and these were first discovered by Kolika scientist. It was initially named as bioplast. The name bioplast was given by Altman, but later the name was changed to mitochondria by Benda. So, these are the scientists who are related with the discovery and naming of mitochondria. Then these mitochondria are mainly present in those cells where the metabolic activity is at a higher rate, where the energy requirement is very high, and the cells which are metabolically less active, in that the distribution of the mitochondria is throughout the cell, but in a metabolically active cell, wherever the energy requirement is more, in those regions the number of mitochondria will be very high. So, let us compare with a dormant or any structure which is present in a dormancy condition. That means dormancy means which is inactive condition, where the number of mitochondria are very less, but whenever a seed is germinating or when a bud is germinating, so it is a very metabolically active function where the energy is required. So, in those structures the number of mitochondria will be very high, or any structure which is undergoing division or the cell division is taking place in a structure. In such structures where the cell division is taking place, there the number of mitochondria will be very high. So, we can say that actively dividing cells or the meristematic cells contains the highest number of mitochondria. Then below or the base of the flagella or at the basal body part, the number of flagella, the number of mitochondria will be very high, because for its movement the energy is required. For the movement of the flagella, energy is required. That is the reason why the number will be more wherever the point of arising of flagella is present. Likewise, wherever the metabolic activities are very high, there the number of mitochondria will be very high. Then what is the structure of the mitochondria? Mitochondria is generally we consider as it is having a sausage shape or rod shaped structure, or nearly cylindrical in structure or thread like organization, which is present in the cytoplasm. Mitochondria is the structure which contains a double membrane nature, or it contains two unit membranes. So, here these two unit membranes are showing some variation in their nature. This is outer membrane which is smooth and without any invaginations anywhere. This is called as a outer unit membrane or outer mitochondrial membrane, whereas inner membrane is showing some foldings towards inner side. So, this inner foldings are called as cristae. Likewise, here this is the inner membrane, and the inner foldings are called as cristae. Outer membrane is permeable to metabolites, why it is permeable to metabolites means, so they contains the membranous porins or aquaporins which will allow the movement of metabolites from outside to inside. But inner membrane is not allowing all type of metabolites into the cell, because it contains restricted membranous organization which will not allow all types of solutes or metabolites into the mitochondrial matrix. So that variation can be seen in the permeable nature in the of outer membrane and the inner membrane. And at some places, okay, actually here we can see some gap present between the outer membrane and inner membrane. This gap is called as perimitochondrial space. Or otherwise we can also call it as a outer space. So this is a space where it is providing some sort of protection to the inner structures. So perimitochondrial space is a structure which is helping in protecting the inner structures, whereas inner space is there. This inner space is called as a mitochondrial matrix. Mitochondrial matrix. So this mitochondrial matrix is a main region or the center where all functions related to the mitochondria are taking place. So of course this is a basic organization that we can see in the mitochondria. So if you see the chemical composition of the mitochondria is made up of proteins which are accounting for 60 to 70%, lipids which are accounting for 25 to 35%, RNA with 5 to 7%, and little amount of DNA and traces of calcium phosphate and magnesium phosphate. So these are the minerals which are present in them. So this is the chemical composition of the mitochondria. Then majority of the proteins and lipids are the part of the membranous structures. Apart from that, some amount of proteins and lipids along with the RNA and DNA, calcium and magnesium phosphates are present in the inner space. Majority of them are present in the inner space. Along with that, the inner space also contains the enzymes are related to Krebs cycle, except succinic dehydrogenase enzyme, because succinic dehydrogenase enzyme is present in the inner membrane. So this is the organization that we can see in here, and on the walls of inner membrane we can see some specialized particles. Those particles are called as oxysomes. So if you see the membrane structures, so this is the outer membrane where we can see the aquaporins, porine structure, whereas this is the inner membrane. With cristae and intracrystal space, that is inside that cristae the space is present, and this is a perimitochondrial space or outer space. This is the outer membrane. So this is the inner membrane. Then these are the porins which are helping in the movement of metabolites from outside to inside. Then this is the cristae which have been formed by the folding like structure. So here on this cristae there are some specialized structures are called as F0 and F1 particles, or we can also call them as oxysomes. Then these are having three parts actually: the base, stalk and the head region, along with the cytochromes, or we can say that electron carriers are present. Likewise, this type of structures are present throughout the length of inner membrane. So if you see the individual structure here on this side also we can see the same type of organization. So throughout the length the same type of organization is present, and if you see each and every oxysome, the number of oxysomes are ranging from 1 into 10 to the power of 4 to 1 into 10 to the power of 5. In each of the mitochondria, the number of oxysomes are ranging from 1 into 10 to the power of 4 to 1 into 10 to the power of 5. And if you see the each and every structure of oxysome, this part is called as F0, or we can all call it as base. This part is called as stalk and head region. So the base will be generally this part is called F1. So this base and stalk are embedded in the matrix region. Sorry, repeat. The F0 part, that is base and the stalk, or both of them are embedded in the cristae, whereas F1 is protruded into the mitochondrial matrix. Then the base is the structure which is having 11 nanometers in its length, that is long, and 1.5 nanometers in its thickness, whereas stalk is a part which is having 5 nanometers in its length and 3.5 nanometers in its broadness, whereas the head region is having 8.5 nanometers in its diameter. This type of organization is there with the F1 particle or F0 F1 particle, which is the structure mainly concerned with the synthesis of ATP. And in the inner membrane, that is in the cristae part, the main function is taking place, that is called as the electron transport system or the oxidative phosphorylation. That is a process where the coenzymes or the NADP like complex energy forms are converted into ATP forms, that is simpler form of energy which is readily available for any type of metabolic functions. So these are concerned with the electron transport system or oxidative phosphorylation, and otherwise these are concerned with the synthesis of ATP from NADP molecules. So this type of structures are present on the cristae which are concerned with the electron transport system. Then the inner part is having the mitochondrial matrix, we call it as the inner space also. Major portion is present which is having the semi-solid like colorless homogeneous material. Along with that all that material, it also contains proteins, lipids, RNA, DNA and calcium phosphate and also the magnesium phosphates. The inner space or mitochondrial matrix. So this is the part which is surrounded by the inner membrane or the cristae. So this part contains various types of enzymes related to Krebs cycle, except succinic dehydrogenase enzyme, and DNA, RNA, various types of inorganic materials are also present. So this is the one where the Krebs cycle is taking place. All the enzymes except succinic dehydrogenase is present. So it is also the one where the various types of amino acids are also present, and intermediate compounds of Krebs cycle are also been formed inside them. the enzymes of Krebs cycle and also the intermediate compounds of enzymes are present in the mitochondrial matrix. So, this is a composition related to mitochondrial matrix, and they also contain the ribosomes, which are of either 55 Swedberg units, or sometimes they may are having ribosomes which are of 70 Swedberg units. So, these are as they contains the ribosomes inside the mitochondrial matrix, the synthesis of proteins is also been taking place, which are produced from the amino acids which are already existing inside the mitochondrial matrix, and those amino acids are produced from the RNA, mRNA, tRNA and rRNA, which are concerned with the protein synthesis. All that mechanism is being operated inside the mitochondrial matrix. Then what is the size of mitochondria? So, as we said that the mitochondria are having specific shape and structure. So, the size of mitochondria ranging from 1 micron to 4.5 micrometers in its length, and 0.2 to 1 microns in its diameter. So, this is the size of mitochondria. Then functions, okay, here mitochondria is called as a semi-autonomous cell organelle. Why it is so called as semi-autonomous? Autonomous means which is performing its functions by itself, but semi-autonomous means some of the functions are performed by itself and some other functions are controlled by the nucleus of the cell in which the mitochondria is existing. As we said that some functions are performed by itself, what is the reason why it is autonomous or autonomy or semi-autonomous in its nature? So, whatever the DNA that is present, this DNA is generally naked, circular, double stranded DNA. So, this is one reason where it is controlling some of the functions of the mitochondria, and this DNA is helping the formation of mRNA, tRNA and also rRNA inside the mitochondria. Then they are also concerned with the conversion of amino acids to form into proteins. So, likewise some of the functions are performed by itself, but it is not that all the functions are under the control of the DNA present inside the mitochondria. Then they also contains the ribosomes, like either 70S type or 55S type. So, but some of the functions, as we said, of course, we said that ribosomes are concerned with the synthesis of proteins because they are the protein factories. And the presence of naked circular double stranded DNA and ribosomes is a characteristic feature of a prokaryotes which are exhibited by the mitochondria. And as they are similar to that of an prokaryotic cell, we can also call mitochondria as a prokaryotic cell within a eukaryotic cell. Then some functions are also been controlled by the nucleus of the cell. So, we call this mitochondria semi-autonomous, or we can also call it as a self-duplicating cell organelle. How does the number of mitochondria will increase in a cell? It is by a process called as fission or division of the mitochondria into the daughter mitochondria by binary fission. Then what are the functions related to mitochondria? Here it is mainly concerned with aerobic respiration. So, during this aerobic respiration, the food materials are completely oxidized to produce carbon dioxide and water. All this process is taking place in different parts of the mitochondria, where the carbohydrates or various types of food materials are completely oxidized. When they are completely oxidized, they are giving rise to carbon dioxide and water. Apart from, they will be releasing energy. So, the energy which is formed is NADH2, or sometimes directly ATP will be formed, and whatever the NADH2 is formed, it is a complex form of energy that have to be converted into ATP. So, here in this process oxidative phosphorylation will be taking place. So, by a process called as electron transport system. Okay, this entire process will be taking place in the inner mitochondrial membrane and oxysomes. So, these oxysomes are acting as ATPase, which will help in the movement of the electrons from one side to the other side of the membrane. So, in this process the NADH2 will undergo oxidation. In this process the electrons will be moving through the various types of electron carriers. So, the electron carriers which are present here in the inner membrane are NADH oxidase, succinate NADH2 oxidoreductase, succinate oxidoreductase. Such type of electron carriers are there which are helping in the movement of the electrons from one side to other side, in which the protons are also being released. So, which is leading to the formation of ATP molecules. So, during the process of oxidation the ATP is formed. So, we call it as a oxidative phosphorylation process. Actually, when NADH is oxidized, so during the process the protons will be moving from one side of the membrane to the other side. So, because of that one the proton accumulation is taking place. So, in order to reduce the proton accumulation, the protons will be moving from the ATPase or the oxysomes. So, in that process when 3 protons are moving, the one ATP will be produced. So, this entire process is called as oxidative phosphorylation, which is taking place in the inner membrane and the oxysomes. As the ATP is synthesized, so wherever the energy is required, ATP is used for various types of metabolic activities. That is the reason why we call this mitochondria as the powerhouse of the cell, which is providing the energy which is required for performing various types of metabolic functions. So, likewise, starting from germination of the seed till the death of the plant, for each and every metabolic function like absorption and transportation of materials, like various functions, the energy is required. For all the functions, for majority of the functions, the energy is provided by the mitochondria. So, we call this as a powerhouse of the cell or biofurnaces.